So it's my pleasure to introduce Fred Hemrick. He is at the University of Maryland, as well as NASA, as far as I'm aware, and is just like an amazing resource for me personally as the other land person on the PACE mission. We've worked a lot together over this past 2 years, I think now, and he's really responsible for the science behind what products I can give you guys as the land team. So with that, take it away, Fred.
Okay. So I'm here to represent land in a very ocean atmosphere environment. So you all know PACE collects data over land. And the thing that is really exciting about PACE to me is that we get repeated global hyperspectral measurements. And this is the thing that really brings PACE to me. As the land people, the reason the land people aren't that interested, have been slow to embrace PACE, let me say that, has been because basically if you ask land remote sensors what they want from a remote sensing product, they want high spatial resolution. That's what they always ask for. And PACE does not have that. But I'm interested in ecosystem dynamics. And so I have from the beginning been willing to give up spatial resolution in order to look at temporal change. And that's kind of what I'm going to talk about. So I probably don't really need to talk a lot about the nature of the PACE products, but I just wanted to take a second for those of you that aren't land remote sensing people. This is a plot showing the kind of a typical green, terrestrial green plant vegetation reflectance. It's of a cornfield down the road here. And the red lines are basically where the PACE land bands are. And so you can see we have a lot of bands in the visible and near infrared, and that's really useful to us because, again, you ocean guys know this, that's where the pigments are, and we're interested in the pigment contents. The bands, the shortwave IR bands are useful to us because they're sensitive to water content in leaves, and that's also an important thing that we're interested in. So this is what we get this is PACE OCI reflectances, 8-day averages for 2 sites. These are 2 sites that I work on. One is the one on the left is the seasonal change in spectral reflectance for a tall grass prairie in Kansas. Excuse me. And the other is the seasonal change in spectral reflectance for a boreal forest in Alaska. And you can see, for example, in the boreal forest example on the right, you can see the really bright reflectance in the early part of the year that drops down. That's snow. So we're seeing snow melt and then basically green up of the vegetation there, which is interesting because it's evergreen forest. And so we can see spectral changes within what is kind of typically continuously green forest. In the tall grass prairie, you're seeing a site that is basically completely dry and dead in the early spring and then greens up like your lawn does. The other thing that, besides the spectral reflectance, the other thing that PACE provides us with is a set of vegetation indices. And we can break them down to 2 groups. There's 10 indices. There are the heritage indices, which are indices that could be produced by MODIS. And these include NDVI and EVI. Those are indices that are generally related to the amount of green leaves in the canopy. Then there's NDWI, NDII. These are indices that are related to water content in the foliage. And then there's the CCI, the chlorophyll carotenoid index, and that's related to basically the fractions of chlorophyll and carotenoid in the foliage. And then the normalized difference snow index, which is a measure of how much snow is on the ground, like snow coverage. I'll just add, CCI is the reason I am here, because when MODIS started up, we were working on trying to look at leaf pigments prior to MODIS. And the band, that 530 band that's in the CCI, is a band that we were really interested in. In MODIS, in the MODIS land bands, they don't have that band. But the MODIS ocean bands does. And so we started processing the ocean data to merge with the land data to get that CCI index. And that started me talking to ocean people and getting involved in this. And that's why when PACE came along, I was looking to work on that. But then what really excites me about PACE is we have these indices that we could not do with MODIS. We can't do with Landsat. And these are indices that generally are related to leaf foliar pigments. So there's a chlorophyll index, and it's related to basically the absorption of light by chlorophyll in the canopy. Carotenoid index, again, related to the fraction of carotenoids in the canopy. And chlorophyll is basically driving photosynthesis. It's the potential for photosynthesis. Carotenoids are pigments that are used for photoprotection, and they also can help enhance photosynthesis. Then there's the anthocyanin reflectance index. Anthocyanins are also useful for photoprotection. And then the photochemical reflectance index, the PRI, which is actually where that CCI came from. This is a hyperspectral version that we basically used MODIS bands to do, and that's what the CCI is. But it is related to xanthophyll cycle pigments, which are used for photoprotection and can be related to photosynthetic light use efficiency. So again, this is just a quick explanation of the pigments again. Chlorophyll is the green pigment in leaves. Carotenoids are yellow pigments. So when you see the leaves change color in the fall or in the early spring when they first start to green up and the leaves look kind of yellowy, that's the carotenoid you're seeing. And then the anthocyanins are red pigments. And again, in the fall when the leaves turn color, you'll see red leaves, and that's the anthocyanin. And those pigments, the carotenoids and anthocyanins, are in the leaves all the time. They're just kind of masked by the chlorophyll, so we don't really see them most of the time. But they're there. So what we can do with these indices now with PACE is we can look at time series of those and look at the dynamics of the system. So this is, again, the example from the Tallgrass Prairie site. There's a little picture, sort of a strip picture of what the site looks like. And the green line on that is the NDVI, and that is kind of normally when we talk about phenology, seasonal change in terrestrial vegetation, we're using NDVI. And you can see I don't have a good way to do this, but you can see how the timing of the green up of the NDVI, this blue line, and then the green line is the chlorophyll index. And you can see that basically in terms of timing, the chlorophyll index is offset from the start of green up for NDVI. The same thing here in the fall during senescence, that the chlorophyll index, and the chlorophyll index, as I'll show you in a minute, is more closely related to productivity than NDVI is. And then finally, this third line that's kind of going in the opposite direction, that's the ratio of the carotenoid index to the chlorophyll index. And if you think about these indices, basically, again, what's driving the chlorophyll index, for example, is the amount of chlorophyll in the leaves and the amount of leaves, the leaf area. And that's the same for the carotenoid index. So when you take that ratio, what you're doing is you're kind of canceling out that leaf area value for the most part, and you're really kind of getting closer to the change in the foliar proportions of carotenoids to chlorophyll. And so you see how the season starts up with relatively high levels of carotenoids, and as the season progresses, we get to lower, more chlorophyll, and then again as the season, as we go into the fall, that reverses. And that ratio is related to the photosynthetic efficiency of the plants, and it's also giving us some hint of stress responses. And then this is the boreal forest example. And once again, you can see how the chlorophyll, the green line, is really quite different than the NDVI in terms of like what green up is. But the thing that's really interesting here is that ratio of carotenoids to chlorophyll. And you see there's a big peak coming up in the spring, and it does another big peak in the fall. And remember, this site, and again, you can see the little picture there on the side, is dominated by white spruce, which are evergreens. And what you're seeing in those peaks there is basically the biochemical changes that go on in these trees as they basically wake up. Their greenness doesn't change a lot, but their pigment contents change as they basically go from being winter hardened to turning on photosynthesis. And we can see those peaks, and they're giving us a hint that that's what's happening in these trees. But we can carry this over now, and this is the great thing about PACE, is we can look at the entire planet with this. And so this is a color, this is just an RGB image of these three indices, green being chlorophyll, red being anthocyanin, and blue being carotenoids. And so basically the colors that you see is the relative difference in these pigments. And the brightness is generally related to how much leaves there are. And so we can look at the dynamics of the entire planet with PACE, and I find this incredibly exciting. And so here's a close-up of of North America, and what I want to point out, and it goes kind of fast here, but as you watch it go through, like look at the eastern US in the spring, you'll see it starts out kind of blue. We'll cycle through a few times here. And then, and that's, again, those being relatively high in carotenoids in the beginning of the season, and then it turns green or white, because white's like high values of all of them. As the chlorophyll comes in, they become productive, and then you see it turn reddish in the fall as you're seeing the anthocyanins start to really come out in the fall as the leaves turn color as we're approaching now. You can see, so you can really see that in the forest, and also then contrast, like look at like in Pennsylvania, eastern part of the country, and the Midwest, which is mainly corn. You know, it's farmland, and you'll see how even though they're at the same latitude, you see very different seasonal patterns in the pigments, because the dynamics of the corn crops is really much simpler than the dynamics of the forests. So we can take this information and quantify it. Hello? Come on. There we go. And so this is basically this is a classification then of the United States based on the dynamics of those pigment indices. And so this is a really simple unsupervised classification. But you can see these different regions coming out. You know, you can see the southern pine forests, the Appalachians. You can see the Corn Belt clearly. You can see the evergreen conifers of the Pacific Northwest. You can see the tropical forests. These come out, and each one of these has with it a defined characteristics of seasonal patterns of these indices. And then, you know, here's like the indices for each, this is for each of 10 of these classes. And you can see how they really are clearly different. And so this is telling us that those different bioregions that are being defined by this classification are in fact have interesting pigment dynamics, and that's telling us something about the physiology of those places. I want to show you just one other example of this. This is the northern part of North America, and again you can see the classifications based on the pigment seasonal pigment patterns. But then what's interesting to me, part of my work is I'm working in the Arctic as part of the NASA Above project. And so the upper classification map there is, again, this pigment dynamics classification based on the seasonal pigment patterns. The lower classification is one based on spectral reflectance from midsummer, from August. And what you see is they're different. I mean, there's some similarities to them. You can see places where they're similar to, but over here around Great Slave Lake, you can see this big blob of yellow here in the spectral reflectance classification, which is actually broken up into a couple of different classes in the seasonal. And so this is telling us that this spectral information from midsummer, and so here's the spectra of the averages of each of these classes. And in fact, what this is these groups that have relatively low reflectance in the red, this is basically predominantly variations due to changes in green leaf area in vascular plants. These ones that are relatively high, which tend to be classes that are over here in the east, look to me like lichen spectral reflectance. And so we're seeing variations in lichen cover here. So two fundamentally different vegetation types are turning up in the spectral reflectance there. Another thing we can do with this is, this is a classification just of this region in the rectangle, and it's just using June, July, and August data. And the idea here is that this is, again, predominantly cropland. And we see that when we do that, again, this is just an unsupervised classification, but we see this big variability in the chlorophyll index and this photochemical reflectance index across these different sites, which is telling us something about management or climate response. I mean, this is my thorn. I don't know what this actually means, but I'm seeing these changes. And we can use this chlorophyll index, which is basically related to the photosynthetic potential. How much chlorophyll there is going to set a limit on how productive the sites can be. And you can see there's a lot of variability in that. And then the PRI, the photochemical reflectance index, is really related to photosynthetic light use efficiency. And so this is, and so again, we see a bunch of differences in there. I have put these together in a really simple way to estimate relative summer productivity of these different classes. And you can see the variability in productivity across all of these different classes. So cluster 10 is this one down here. So this is a far western cluster, and it's not surprising that this is a fairly low productivity. And over here, cluster 9, one of the easternmost clusters, this is one of the more productive ones. We can see the difference in the productivity from that. One other thing I just want to mention, this is almost more for fun, but one of the things that we can do as we come into fall is we can use these indices to see fall colors emerge in the forest. And so here in the scatter plot is showing the scatter plot is basically points from a region around in here in New Hampshire and Vermont. And it's showing the chlorophyll index versus the anthocyanin index. And what you see is in late summer, we have high chlorophyll, low anthocyanins. And as we move into fall, the amount of anthocyanins increase again. And then what we see is chlorophyll, the anthocyanins stay constant, but the chlorophyll decreases. So if we look at the slope of these different clusters, so basically anthocyanin over chlorophyll, we can map out where we think the leaves are turning red. And this kind of matches up with, this is an estimate, so this is from last fall, and this is an estimate around the same time based on a model using environmental data to describe it. And you can see that where we're seeing, here in the Adirondacks and the White Mountains, you can see that those are occurring in here. Again, my thorn is I don't have a lot of ground data for that, but one of the sources of ground data I can use are phenocams. I don't know if you guys are familiar with phenocams, but there is a whole network of web cameras mounted on various places all around the world. And basically, they record a photograph every half hour or so. And you can take those images, you can define an area within the image and pull out the red, green, and blue bands from that just color image. And you can calculate, well, what they normally calculate is what's called a green chromatic coordinate, which is green, the green value over the sum of the red, green, and blue. Because I'm interested in trying to get fall color, I did a red coordinate, where it's the red over the sum, and that's this red line for this site. This is a site in New Hampshire, and that's the red line, and the black line here is that ratio of the anthocyanin index to the chlorophyll index. And so there's some hint here that we can detect the color change in the leaves. But we probably really want to do something more quantitative, and so one of the other things we've been doing is looking at ecosystem productivity from PACE for terrestrial things. So the spectral reflectance of vegetation is related to the structure of the canopy, how much leaf area there is, how clumpy it is, which control really how much light the leaves intercept. And then there are other characteristics involving chlorophyll content and these other pigments, which relate to how much photosynthesis there is, how much potential photosynthesis is, and photosynthetic downregulation. All of those factors, those physiological factors, also affect both the spectral reflectance and the productivity of the plant. So we have like a conceptual idea that we can link spectral reflectance and productivity. Now there is a MODIS gross primary productivity product that exists, but that product uses weather data, humidity and temperature, to help define the GPP. And what I'm doing here with the PACE data is taking advantage of the PACE spectral data to do this only with reflectance. So instead of looking at the weather data and having it tell you how stressed the plants are, or how the plants are performing, I am looking at the spectral reflectance, having the plants tell me how they're performing. So to do this, I've been working with data from NEON, and again, I don't know if you people are familiar with NEON. NEON's a National Ecological Observatory Network, and it's been established by the National Science Foundation. Basically, it's funded out of the same pot in the National Science Foundation that builds these giant observatories. Well, the ecologists got together and got them to say, well, we'll make an ecological observatory. And so they have divided the country up into 20 different ecoclimatic regions. And within each of those ecoclimatic regions, they have established study sites where they measure all sorts of things, species content. They do a lot of stream water measurements. But for my purposes, they also measure carbon dioxide fluxes. And they do that with these flux towers. This is a picture of one of the flux towers. And so, again, I don't know if you're familiar with eddy covariance as a technique, but it's a really powerful technique for looking at ecosystem processes. And basically the way it works is they've got a device called a sonic anemometer, which in that photograph there are the claw-like things there. And what they do, what the sonic anemometer does is it sends pulses of sound between those probes of those claws. And by looking at the timing, the speed of sound across that gap is affected by the wind speed across that gap. And so they can measure very accurately the wind speed and very accurately the wind speed across that gap, and they can do it like 50 times a second. And so they have these very accurate measurements of the dynamics of the wind speed. And then at the same time, they also have devices that measure the amount of carbon dioxide and moisture in the air by, these are infrared gas analyzers. So basically they suck air through a tube and they shine an IR beam through that, and the attenuation of that IR beam tells you how much water vapor or CO2 is in that. So again, they can do that really, really fast. They can put these things together and add them up and basically calculate fluxes. So as part of NEON, they have 47 of these flux towers, and they have been established in all of these ecoregions they divided the country up into, and really represent, and they've been put in place to really represent the breadth of vegetation types within the United States, from tropical forests to Arctic tundra. And so what I've done with that data is I have taken the flux tower data, calculated gross primary productivity from that, averaged it to 8-day averages to match up with the 8-day average PACE data I was working with, extracted, well, I didn't do it, Sky extracted the PACE reflectances for the 4-kilometer pixels for each of these towers, and we matched them up. And this gives us over 1,000 observations to work with then. And just to show you what an advanced PACE has been, prior to PACE, I was doing exactly the same kind of research, but I was using a different satellite, DESIS, to do this, and I'd worked really, really hard on that for a while, and I had 12 sites and 70-some points. So after years of work, and so this is like in less than 1 year, I had over 1,000 points and 47 sites to work with. So this is the qualitative difference that PACE makes. Hello, come on, there we go. So let's start with something simple. We can take chlorophyll index. Again, we talked about how chlorophyll is represents the potential photosynthesis. And again, I've just thrown all of these different sites together into the analysis and just plotted them all up. And what you see is, it describes 66% of the variance across all of these sites. So, oh, and again, this is 47 sites, and we're looking at data from March through October. So we're looking at a wide variety of vegetation types and a seasonal change here. And so, but we're describing 66% of the variance with a RMSE of a little less than 5. That's really pretty good. But the other thing that we can see here is if you look at the residuals, what you find is a lot of the points with large residuals on this turn out to be the agricultural sites. And so that may well be because they are managed in ways that the natural sites aren't fertilized. And it may also have to do with the fact that generally the plots with these agricultural, that the flux tower is in and the agricultural sites are relatively small compared to the 4-kilometer size of the PACE pixel. So that might be another factor. But if you remove those points, the R squared goes up to 0.74. So now we're getting really something pretty good. But we can do better than that because we've got all these other spectral bands. So here, what I've done is I've done a partial least squares regression on all of these bands and got this result. Now, for all of the points, it's an R squared of 0.74, and if you take out the agricultural sites, it goes up to 0.8. So, we're getting really pretty good results here. And the thing I like about partially squares regression is what you get out of it is you get a coefficient for every spectral band, and that's what this plot is. It's showing and we can look at that because we know something about how spectral reflectance is, the significance of them. We can say something about what's driving productivity across North America. And so we see this big spikes here because basically the magnitude of the coefficient is going to say which ones are most important. And we see this here, and this is that red edge, that transition from the high absorption in the visible into the low absorption in the near infrared, and that's strongly related to chlorophyll content. And so this is basically a note, this is chlorophyll. This little peak out here in the blue is also on the edge of the chlorophyll absorption band for the blue, and that's also probably another chlorophyll absorption thing. These large values out here in the shortwave IR are related to foliar water content. And again, foliar water content is really important. And then finally, this little tiny peak right here, that one, is the band that is used in the PRI, the photochemical reflectance index, and the CCI. And so even though we don't have PRI in this model, the results from it is telling us that it has some significance. And so then you can see, we can take those coefficients and then apply them across all of North America. And you can see the seasonal change in productivity across North America with this. But we can do even better than that if we train more locally instead of drawing all of the points in together. And in this case, what I've done is I've just broken it out by those ecoregions, the 20 different ecoregions, and trained each one of those separately. And now, now we're getting overall an R-squared of 0.88, which is just and a root mean square error of less than 3. And so this is really, really good. The problem with this is that and the other thing to say is when you do that kind of training, now those agricultural sites fall in line because they've been specifically trained on. And in fact, you can see it. So here are the coefficients now for all of those different regions. And this blue one that's really weird, that's one of the agricultural sites. So you can see what's going on. But the problem with this is that look at how variable these coefficients are. So what that means is if we were trying to carry this over to continental or global scales, we have to do some sort of classification and pick which set of these coefficients we're going to use. And if we get it wrong, we could be very, very wrong. And what is the optimum way to do this? I don't know. There's another thorn. But if you do know what site you're working at and you have it trained properly, you could do really, really well. So this is the Guanica dry forest in Puerto Rico. And again, this is time. And the orange line with the error bars is the gross primary productivity is measured on the flux tower there. The blue line is the estimated productivity from PACE. So you can see it's doing a really good job of capturing this variability. So basically it's dry, it gets some rain, it's not greening up, it just photosynthetically turns on, and then it drops down, dries out again, comes back up again. It does this whole pattern. The green line is NDVI, which is typically what's used to monitor vegetation. And you'll see that NDVI is not capturing this variability that we're getting here, but we're able to describe that with the PACE data. And then because the agricultural sites were weird, if we train on the agricultural sites, we can get good results. And again, so here is this is a tropical agricultural site in Puerto Rico, and it also is doing this variability because it's multiple cropping. And once again, the green is the GPP from the flux tower, and the black is the PACE estimation. This is the Konza Prairie. This is a site in Kansas, mainly corn, I think, that year. And again, we can see that the PACE data is tracking this really well. It does a pretty good agreement here. But once again, if you look at the coefficients, they're quite different. And so that's, again, a problem that we need to be able to understand what drives these differences and how to use them, how to best use those to get the best results. So I won't end with conclusion. I'll end with some challenges to you guys. How can we... So one of the challenges that we have is, I've been showing these pigment indices, and basically I've fallen back on the pigment indices because they're an easy tool to use. We have some idea what they mean, and we can easily look at them over time. But they're probably, and the other thing to say about it is prior to PACE being launched, the number of data sets that we had that had repeated seasonal measurements of spectral reflectance of terrestrial surfaces, or like literally a handful of them. There's very, very few of them, and they're very localized because it takes a huge amount of work either for someone to go out on the ground and measure that all the time, or very expensive if you're going to fly an airplane multiple times. And so we just don't have very many data sets. So the whole idea of what's the best way to take a time series of spectral data and extract useful information out of it, taking into account both the temporal and the spectral characteristics, is something that really has not been explored very well, and is the kind of thing that really work needs to be done on to find efficient ways to make use of that information. And then on top of that is, again, we can look at these indices, and the indices just use a couple of spectral bands to relate to some variable, and you can kind of look at them. But are those the best spectral bands? All the work that's been done on those has been like in cornfields. And so, again, we don't have global measurements of these. And so what are the best spectral bands to get out information on change in ecosystems? That's something that's, again, we don't know, and it's just it's another thorn. My glasses aren't that good, I need to read it here. And then can we identify spectral changes associated with stress responses? So one of the things I'm interested in doing this year, so we've done that productivity estimate, you've seen that, but what I want to do is look at time series changes, time series between like this year and last year, to see if, do they track the same? Are they different? And if they're different, can we associate stress responses to these based on, was there a drought? Was there a heat wave? Can we see spectral changes associated with these kinds of environmental events? And again, this is an open topic. And then one of the other problems I've been having in doing this work is when you look at these time series of the data, I've been looking generally at 8-day averages, and you'll look at an 8-day average and there'll be like a drop, and then it'll go back up again, and you'll, like in the chlorophyll index, and you're like, well, is that because there was an actual change in chlorophyll, or is this just like some bad data? Is there some sort of problem with the data processing? And identifying the difference between problems with the atmospheric correction or angular views or other factors and actual stress responses is something that needs to be worked out. And then again, what's the best way to describe this spectral change over time? How do we do that? And then if we can identify these responses due to stress, can we diagnose those? Do different types of stress responses, like is response to drought different than the response to heat? Can we see those, and do those have different spectral responses in terms of, that we can detect? So if they do, then we would be able to look at the spectra from PACE and say something like, this region is drought stressed, this region is heat stressed. And so it would allow us to diagnose what the plants are doing. And then on top of that, then the other side of it is, do different vegetation types have different kinds of responses to stresses? So, do pine trees look different under heat stress than oak trees? And that would be it. And then, as I said before, the other challenge here is to improve the GPP retrievals. How do we segment the world to get the best results? And that's a question. And I can tell you that, like in the data that I've been working with the NEON data, I have separated out, for example, all of the deciduous forest sites, and hoping that, all of those parameters are, coefficients are going to look the same, and they don't. So I don't know how to divide up the landscape. And then again, if we do detect stress events, are there spectral changes affiliated with like early onset of stress that we could use to detect stress before before something gets really bad? For example, insect infestations, or for example, the indication that the forest is becoming stressed, which may mean it is more likely to catch fire or have an insect infestation. So these are all things I believe that can be done with PACE, but are out there to do. and go and do them. And I just want to thank NEON for giving me data to work with, because without that we wouldn't be able to do this. So thank you. Any questions?
Questions, yeah.
Thanks. That last figure you showed before this one, where you had the plot of the spectral variance, I know you said you don't have all the answers, but what would cause the majority of the variance to be explained in the visible as opposed to, let's say, the near infrared? Because I know most appear to be really right in the near IR.
Well, this part here,
Yeah.
this is all driven by chlorophyll. This is the chlorophyll red edge here. Mm-hmm. And it's just that, for reasons I don't understand, this tropical forest just has wildly huge effects of, well, actually I do know, because this is basically a dry tropical area. And so what's happening is the leaves dry out, and that's a strong signal of photosynthetic ability in these sites. And so that's what, so this is shortwave IR, so these are affected by leaf water content.
Okay. And so a lot of those plots, right, there's a lot going on in the near IR, and just past 1,000, that's all like water effects?
Yeah, basically it's water, and the two things that really drive the reflectance in the shortwave is leaf water content and overall leaf area.
Yeah, thank you.
Yeah.
I have like so many questions. I'm going to try to narrow it down, because just the ocean is notoriously horrendous at doing productivity in terms of, agreement between models, and so I'm like on the edge of my seat kind of seeing how things are done in another field here. But in the ocean, we're always concerned with net primary productivity after the respiration effects, essentially representing the amount of total energy that's sort of available to the system. I imagine these systems are able to measure the CO2 flux in the evening as well.
Yeah,
Is
so
respiration
I
important
skipped over that. What you really have to do is the eddy covariance system measures net ecosystem exchange. So it's measuring the net CO2, and that breaks down into respiration and photosynthesis. And what I did, there's several ways of doing this, but the partition what I did is you take nighttime data when there is no photosynthesis, and you develop a relationship between nighttime respiration and air temperature, and then you apply that during the daytime, and that's basically how you, and then so you subtract out the respiration part, and you just get the photosynthesis part.
Ah, okay. So that's such different. We use different terms in the ocean. GPP is all. So the GPP in this sense has already had the respiration component. I don't know why the communities don't talk more. It's just incredibly
Actually,
confusing.
we've been talking that because every time I talk to the ocean groups, like the ocean people are going, pigments, yes, we are so into pigments. And I'm going, yeah. So it's like, we really need to have a meeting of like the terrestrial ecologists and the oceanographers to talk this over, because they're all plants. I mean, I'm able to glom on to PACE because the ocean people are interested in these pigments. I'm interested in these pigments. And the pigments have spectral characteristics. And so the ocean picks them up. You guys pick them up in the ocean. And I can just waltz right in and say, yeah, I'll take that data. Thank you very much.
Isn't this the best mission to do that on?
Just curious, when observing the canopy, the directionality affects the derivation of the vegetation indices.
Pardon me? So what?
Like when observing the canopies,
yes.
The directionality affects
its
derivation.
So this is a serious problem in PACE, is that there are strong view angle effects on terrestrial vegetation reflectance. And this is why everything I do, which are not corrected for, sky. But we have schemes in mind to do that, to correct a nadir reflectance. But that's in fact why I work with the 8-day averages, because my thinking here is, and it's really sort of hoping, is that by averaging these different view angles together, it kind of blurs out the effect, at least biases due to angular effects, and so you can kind of pretend they're not there. And that's pretty much how I go through life. I just pretend they're not there. But they are there, and they are a serious issue.
Yeah, because I'm thinking the simultaneous measurement from the polarimetries.
Yes.
Kind of offering some extra information.
Yeah, so the thing, so I'll say two things about this. So yes, one thing that could really be useful is to take the bidirectional reflectance functions you can derive from the polarimeter data and apply that to do the corrections for the to nadir view of the OCI data. So that's one thing you can do. But the other thing you can do with this, which is to me more interesting, is that this PRI index that is related to that's related to photosynthetic light use efficiency, it's strongly affected by basically chemically in the plants. It's strongly affected by the amount of light on the canopy. And one way to really tease out what it's exactly doing is getting a bidirectional view of PRI. So that because if you're looking from one direction, think about a forest. You're looking from one direction, you're seeing a lot of shaded leaves if you're looking towards the sun. So you're seeing the shaded side of the tree canopies, and that is generally not very stressed because light really stresses the plants. So that's not very stressed. So you can get a PRI of relatively unstressed leaves. But then you go around to the other side and you're looking at the illuminated side of the canopy. Then what you're doing is you're seeing the leaves that are most stressed because they're the ones that are most illuminated. And you can take that difference and use that to basically solve for a better solution for light use efficiency, photosynthetic light use efficiency. And a few years ago, colleagues of mine were proposing a satellite to do exactly that. And they've retired now, but I was just commenting to them that specs could do this. You could do this with specs. And so that's so that's an interesting, there's an interesting project there.
Thanks.
Thank you so much. Even though you asked about BRDF, which is my least favorite topic. Kieran, did you have it?
Have you considered using a weighted numerical optimization for the PSLR coefficients?
Nope. A, I'm really dumb. I just use the simplest thing I can possibly do. And B, my whole strategy with, I'll be completely honest with you, my whole strategy with PACE right now is I am just skimming through here and trying to do what I can the easiest way I can. And I'm expecting people to come along behind me. And because basically, I kind of see a lot of my goal as kind of the only land guy in PACE. I'm not the only land guy in PACE anymore, but I'm going to say that anyway. As the land guy in PACE, I see one of my jobs is to convince people that you should use PACE. So I do these kinds of things, quick and dirty, to say, look, there's something here, and hopefully somebody who is much better at this stuff than I am will come along and actually do it. So you guys go do that, because you're all better at this stuff than I am.
I'll send you an email. I have the code, so.
No. Become a multidisciplinary person.
That's right. You don't have to stay in the ocean. You can come on land.
Coasts.